Hi, in this video I am going to go through and I am going to make some cohorts through the GDC data portal. This is a database that we'll be using in this course in order to explore some authentic cancer genomic data sets. In particular, we're going to be using them to make these cohorts. These are like groups of different patients that allow us to do a differential expression analysis. And that just being where we compare the gene expression as measured by the amount of RNA that's being produced by their cells from one group to another. So we're doing a very simple form of differential expression where we're just targeting two groups, two fairly well controlled. So the ideal is that they're very comparable, very similar tissues, very similar situations, except there's one small difference. And that small difference is ideally what we're focused on within the differential expression analysis, where we're trying to analyze, well, which things are different between them. And those are usually going to be in correlation with some sort of interesting pathway or interesting condition that is true about those samples. So common examples are going to be things like taking for a particular type of tissue, a particular type of cancer, maybe even like a particular gender, and comparing people with all those controls, but with different stages of that particular cancer, and trying to see what genes are associated with the difference between those stages of cancer. And what we'll see is that there are certain genes that are commonly associated with like the transition from stage 2 to stage 3, or from stage 3 to stage 4, as the cells start to become metastatic and travel around the body. There are certain gene pathways that just tend to be upregulated. Those are going to be a lot of your oncogenes, genes that promote cancer through their expression. So imagining cell genes like the things that tell the cell to replicate. While they get upregulated, they get produced more in cells that are more cancerous. And genes that are downregulated in cells that are more cancerous are usually things like our tumor suppressor genes, which again makes sense that they would be produced less. Additionally, there's a lot of kind of just correlated genes that we don't tend to think about. These are going to be genes like rearranging cell structures and changing the surface morphology that are very commonly differentially expressed in these cancer data sets. Even if they themselves are not the cause, they are correlated. And that's what this analysis finds. But to do that, we need to create some cohorts. So let's go get into it. All right, here's your database. There's a lot of details here. You should absolutely be reading this. You are agreeing to use this database for its intended purpose, which is for research. You are not going to use it to persecute anybody. There are people very kindly generated their or donated their data when they were very sick, and I think we should always be respectful of that. Yeah. All right, so I'm going to click accept. I'm going to click dismiss on some of these banners, so we're going to find out that today, actually, when I'm recording this, it may be shutting down for a bit, which we'll see, because I don't know if that's going to happen because of this other banner, which I believe will go away, and if not, I'll show you how we can hide it, at least for right now. We're going to start building a cohort, so that's a collection of cases through the cohort builder. And this does look like it's not going to go away. So I'm going to try to block it using what is that? It is called, oh my gosh, why won't you say your name? Don't you want me to be able to advertise for you? uBlock, I think. Yeah, uBlock Origin. This extension for Firefox, for me at least, allows me to block the element. There we go. So I've just created a block, and now it's gone away. Lovely. Okay, so moving forward, now let's try to create some grouping. So you can see on my computer, I've created a few before, but I'm going to erase those just so we're starting fresh. And now that I've done that, this number of cases up here has gone way up. So this is basically the total amount of patients that have been characterized. I mean, if you can imagine, each one of these sequencing runs, it is costing at least hundreds of dollars. This is a huge amount of money that has been invested in this data set. And it's why we're using this database in my class. It's a tremendous resource. Now, that said, I do restrict my classroom to a particular subset of the data that I've come to get familiar with over the years, and I just kind of know some of the quirks. That's one of the things you're going to learn is whenever you're working with real data, working with real research, there's a lot of data cleaning that has to occur. And I've done some for you, like I'm heading off, like in the background, I'm heading off certain problems that you might run into. And since if you use a different program, um, I might not know those issues, I'm restricting you to the TCGA program. So that was a particular program. Now, we'll note it is the it used to be the biggest, but it's now the second biggest data set in here. But it's comprised of all sorts of different cancers. So you can look up what these acronyms are, but they're different types of cancer. Additionally, the initial kind of subsetting. So right off the bat, we've gone down from 45,000 to 11,000. But that turns out to not be sufficient because we're doing differential expression analysis. And not all of the patients had differential expression data collected. So that would just be gene expression data. The difference comes when we compare them. But the gene expression data is what's used for differential expression. So to do that, we're going to need to come down to available data. We're going to need to check to see, first of all, do I have access to it? I don't have access to all of the cases data. So we're going to select open access. Additionally, there's lots of different data types within this. The type that is used in differential expression would actually be the gene expression. Kind of new is, oh, I'm not seeing it. If you see other things on here that sound like differential expression, it's usually downstream analyses. I'm not seeing it, or maybe it's because they're not open access. That might have been what it was. We'll see if I turn that off, if it appears again. No. Oh, there it is. We have like isoform expression. We have some other expression here. There's a lot of related analyses, but what we're looking for is the gene expression quantification. Specifically, we're looking for the star counts, which may or may not always be necessary. There are other ways of calculating how this data should be collected. But this is kind of like minimum, or this is kind of redundant partially, so I think I can take that off. And there is no change because for data that is star counts, that's the only type of data it is. So the workflow star data type, technically redundant. So that's actually sufficient. So from that, I'm going to save my cohort. Now, I wish I had looked up everything that I'm naming these, but I'm just going to try to get pretty close. So star data. So this is me saving that cohort. Now this is going to be saved to a cookie on my computer. I'm not logged in, and that's because it requires privileged access to log in. And while I could probably request it, considering my position, not all of you would be able to get in, and it would be a whole thing. So we're just using the publicly available data, at least for my project. All right, now at this point, I'm going to do something a little bit weird. I'm going to actually make a new cohort instead of simply picking additional workflows. And here's what's going to happen. Later on, I'm going to want to make some decisions about these cases. I'm going to want to group them based on sequencing information. I'm going to want to say, like, all right, group A, you have this gene that that's commonly associated with rectal cancer or colon cancer. And it's intact. Like, you got a good version of that gene. So that's probably not why you developed rectal cancer. And then group 2, you have a high impact mutation in that gene. So that's possibly why actually you developed rectal cancer. And so then I'm going to have these two different groups and I'm going to be able to compare them to each other. However, in order to do that, I have to know whether or not that gene APC was mutated or not. And so to do that, I have to have sequenced that gene and I have to have that data available for me to even evaluate. And so along those lines, I'm going to keep these other two filters in place, but I'm going to choose mass somatic mutations, and there's no kind of subtypes of workflow, so I don't actually need to select anything else. And I'm going to save this as a new cohort. Now please note, I didn't just say save. That would have overwritten my star data. Instead, I'm going to save a new one as my SSM. So that's simple somatic mutations. Now you may note this says mass somatic mutations. It is sometimes referred to as simple somatic mutations, which is why I'm calling it that. Here it's masked because it turns out not all of the genes are publicly available for a lot of reasons, but I mean, one of those is that it includes things like sex chromosomes, and it includes things like heritable genes that are you know, able to really track down like the pedigree of a person. Sorry, I'm a plant breeder by training, so I shouldn't use the word pedigree for humans. The ancestry, there we go, of the different patients. And so there's certain mutations that have been blocked out. Just to, I mean, while it doesn't actually work fully, it does help prevent some of the more abusable markers from being present in the data set, which, you know, I think that's valid. It's to protect the people. And I believe you can get access to more privileged information if you can, you know, produce your credentials. All right, so that's simple somatic mutations, and we're going to do one more data set. It's going to turn out not to be necessary for this particular gene. However, a lot of cancer genes go through what's known as a copy number variant. And so these are where there's like deletion events or duplication events that change how many copies of that gene are present. So they don't show up necessarily in the simple somatic mutations because they're not simple. They're usually big deletions. They can be arms of chromosomes. They can be dozens of genes. They can be a lot. On the other side, you can duplicate entire arms of chromosomes. Cancer cells are wild, and in fact, they're not even really homogeneous. They're commonly these weird kind of heterogeneous clusters of cells where some cells have the right number of genes and some cells have entire extra copies of chromosomes, which normally isn't survivable in a human, but on like a single tumor basis where you don't have as complex structures as organs, the cells might survive for a while. And that's why they survive. They start to get a little bit out of control, and eventually they start to cause problems because, well, it wasn't supposed to have that many chromosomes, and having that many chromosomes turns out to be a problem. So anyway, these copy number variants are fairly common because of that. And so I'm going to call this CNV for copy number variant. And just to refresh what we have 11,000, this is fall of 2025 for the record. So 11,044 CNVs, 10,511 star data, so a lot of data there, and 10,190 for the simple somatic mutation. So each of those were a different number of cases. I wanted to demonstrate why we couldn't just select all three at once. Where's the last one? Gene expression quantification. So if I select all three at once, you'll notice that this number has gone up. So this is now bigger than any of the previous values. And the problem with that is that 11,260 is only possible because these are currently joined by an OR. We don't need an OR, we need an AND. So the AND is not possible through this current view. To get an AND, we're going to actually have to use a separate tool. So I don't want an OR, I want an AND. So let me just reset that. This is what I forgot to do last time. and now we're back to 10,190. If I didn't trust what I had just done, I could have, I believe it's this button, discard changes, which will just revert me back to the date I was before, which is the same, but it just makes sure that I didn't make any changes. All right, so now I'm going to go to the Analysis Center, and we're going to show how to do that and. So I went a little bit too fast. I'm going to click on Set Operations. Set Operations allows me to do set operations. This is a term from set theory, which is just basically how can we compare groups of things and do logic. You also see this referred to as Boolean logic, and there's probably some other terms too that I'm not as familiar with, but well, it's easier to show than to tell. So I've selected all three, and it's going to generate this Venn diagram that breaks down what all three of our different groups look like. And they look like this because, well, for say 307, this is the samples in S1 minus any of the overlap between S2 and S3. So it is in S1, but it is not in S3, and it is not in S2. The 719 is in S1 and it is in S2, so it's the intersection of S1 and S2, but it's not in S3. So intersection of S1 and S2 minus S3. I guess these are everything that's in at least 2 data sets. None of these are the ones I actually want. In the end, what I'm trying to do is I'm trying to get the samples that are present in all 3. And so this is the group I want. So we can come over here. I could click on this and it checks that box for me. And if I wanted to get something fancy, I could select all 3 that I wanted, if those were the ones I wanted, and then select down here. But it ends up, all the ones that I want are these right here, where they are present in all 3 data sets. So 9,636, at least in this year. Now that's me double checking my numbers. My numbers are currently good. All right, so I'm just going to label this TCGA count them C and D. So this is just telling myself in the future that this is data that has all 3. And I'm just going to go double check how I named it in your notes. And in your notes, I actually labeled it star. So star is the algorithm that's being used to calculate the count. But that is just count data. A lot of acronyms, right? Okay. So we now have kind of a starting point. And at this point, I can really start to do biology. So I'm going to give you like kind of the full tour of how I would do something like this. So let's say I was just going into this. I wanted to look up the projects. If I didn't know what these were, I could just do a quick search and I could be like TCGA project abbreviations. And as I looked them up, I could see that, you know, maybe there's different types that I'm interested in. So like maybe I'm interested in colon cancer. And so I could come over here and I could pick colon cancer project. Once I get within that, I can see that there's actually different types of diseases that have been diagnosed. The biggest one is adenomas and adenocarcinomas. Those are going to be benign and carcinogenic tumors. And so let's pick that, just that way we're not comparing different situations to different situations. Over here, I can see that there are some other primary diagnosis information. And so, you know, I might want to do some filtering here. However, we can see that there was a lot of like not reported values and also a lot of not otherwise specified values. Whenever you see that, it's just because the doctors just didn't, you know, get more detailed diagnostic information. They just didn't provide that information, so not otherwise specified. Oftentimes, when that's the case, I may avoid selecting within that filter if I feel like that data set is not very reliable. And because we're working with humans. I mean, humans are going to be classifying things in all sorts of different ways. Okay, but for right now, we've got these cases. That's our starting point. That's the people that have all of this the different three data sets. I've selected colorectal cancer and I've selected adenomas and adenocarcinomas. I could look through a little bit more of this and I can see, well, the colon, as opposed to the rectosigmoid junction, which is a different part of your rectum, I should probably at least subset it there. One of the biggest issues that you will find in this type of analysis is that it's very sensitive to tissue. That tissue, being a type of cell in a given organ, has a distinct genetic profile fairly commonly. And so you often want to be very specific. Like you definitely don't want to be comparing blood to colon cancer, to colon cells. Even if they're from people with colorectal cancer, they're going to give you massively different RNA profiles. Okay, but let's pretend we're still trying to evaluate. We don't already know what my filters are. I do know because this is an example I've been using for a couple of years now. But let's go look at some of these mutations. So mutation frequency is a great tool for exploring what mutations are available across your present cohort. So please note, right now I have this starting cohort, but then I've added some additional filters to it. So 356 possible cases. When I come down here, I can see that it turns out that the most commonly mutated gene across these cases is a gene called APC. Well, it turns out APC is a gene that is very commonly associated with colorectal cancer. It is an antagonist of the WNT signaling pathway. In other words, it's a transcription regulator. It's involved in a lot of processes, which I don't know how many of you this will scream to, but those to me scream cancer related. Cell migration, adhesion, transcription changes, and apoptosis. Oh boy, we know that's going to be involved in cancer at that point. And indeed, it's a tumor suppressor protein. Defects in this gene cause familial polyposis, which I don't actually know what that is. Again, plant biologist. But we can see then they do a nice little breakdown. It is an autosomal dominant pre-malignant disease that usually progresses to malignancy, and in that case, associated with rectal cancer. So none of this is surprising given what it does within the cell. This is a good target for an analysis. So I could choose this gene and I could start to explore it, and I could say, all right, well, I'm going to be interested in the people who are mutated. Now, when I look at that, it turns out not all mutations are the same. And so I could start to explore through here and I could say, like, all right, well, what if I take only the mutations that I expect to be of high impact? And what we'll see is this organization may change. APC may no longer be at the top once it finishes refreshing. Oh, it still is. We can see that for 254, they actually have a high impact mutation. And indeed, some of those are copy number variant amplifications, and some of those are copy number variant homozygous deletions. So those are all interesting. It means that I have good numbers. In fact, so many that I probably want to trim it down a little bit more. I can do some better controls of this data. So what I could do is I could, usually if I were you, I would actually do some of the downstream steps to isolate the people who lack this mutation, because I can already tell that that's going to be the smaller group, is people who have no mutation in APC, and then trim it from there. But for the sake of this demo, I'm going to just go in and do a little bit of filtering on my initial cohort. So instead of looking for just these, I'm going to get a little bit more specific. So I'm going to come over here. So at this point, we can then filter based on gender. Gender is a common filter to apply, not because it's necessarily always needed, but because it does help avoid certain issues that occur when you have unbalanced selections. So for example, if I ended up with one group with only female patients or cases, and then I had the other group be either a mixture of female or male, or at a much worse scenario where it's just male, what I would end up getting for my differential expression analysis is basically, well, did you have two X chromosomes or did you have a Y chromosome? And that will just be associated with like a handful. Actually, it's not that many, but it's a handful of very differentially expressed genes. Now, because we're going to have filters applied to the sex chromosome, we often won't see the SRY gene present within the male samples. I don't think, at least in past years, they've censored that. But we will often see genes like XIST, which is the non-coding RNA that deactivates the second or more X chromosomes present within female cells or like Klinefelter cells. But anybody who has multiple copies of the X chromosome will produce a lot of this XIST, and it's like present at huge quantities because it's a non-coding RNA that physically interacts with the X chromosome, which is just wild to me. But it's orders of magnitude, just hundreds of orders of magnitude of significance between the two. And so they just kind of cause issues during the analysis process. It's a problem. An easy solution is, and kind of a blunt force solution, is just to use gender as a control. Not necessary in all cases, but sometimes quite helpful. Okay, additional filters. Pathology is often a good choice. Now, not every type of cancer has the same way of categorizing the stage. For rectal cancer, the common one is the AJCC pathologic stage. So these are just grouped into different numbers. So I'm going to pick kind of middle of the road because, well, frankly, it's got the most data in it. I'm going to pick stage 2. However, we'll note that stage 2 is actually an umbrella term and there are subdivisions. So sometimes the doctor has gotten more specific and sometimes they have not. There's also like a 2B, which I currently am not seeing because of my gender filter, but that's also common. As I select these, certain things get hidden from me as they're no longer available. Okay, now at this point, my number's good. I would actually prefer to move forward, but we should always go check out that tissue. And I just really recommend this because your biggest factor in terms of gene expression is what the tissue is. That's the single biggest thing by far. Different tissues express different genes. And so as I look through here, we can see that there is the ascending column, that's like the upper section of the column. There is the cecum, that's the most upper section of the column. Hepatic flexure, I guess that's on the side that's closer to the liver, I guess. I don't know. Sigmoid colon, just different sections of the colon. I'm going to end up picking this ascending and cecum, which if I pulled up a diagram are near each other. They're near the top side of the colon. I'm not going to choose not otherwise specified, so this NOS is not otherwise specified because the colon's kind of a big organ, and it's reasonable that different sections might have different cellular profiles as they're absorbing different things, and they have different issues that they have to encounter, and moisture levels, and I don't know, but I'm going to restrict it to certain tissues. And like this approach of me basically just kind of trying out this tissue is why this is in actuality a fairly hard technique to apply. You have to get a lot of experience, and you know, some of my experience says I should probably be sticking to like a certain section within that organ. Different organs, different situations. I can't give you a one size fits all, unfortunately. Usually though, tissue is a pretty good choice in terms of what should I restrict as a variable. Okay, so at this point, I'm going to just call this a group called coad filtered. I actually saved this as my third take. Ugh. And so I'm going to save this as overriding my previous coad filtered. But just to recap, I started with those 3 combined data sets. I've added on different filters. I took a little bit of a break. I went and analyzed some of the genes just to make sure like I had something I was targeting. I could go back to gene frequency, but I want to save it before moving forward. We can always just keep spinning off newer and newer cohorts, and you can kind of explore it that way. But this is going to be my base. This is where I'm going to select everything else from. Okay, now that I've got that saved, I'm going to go over to mutation frequency. I'm going to wait for it to load. And just to give you a little bit of a tour while we're waiting, oh, hey, we got it there. But still to give you that tour, we have our selection up here. So we're currently at 28 cases. Right now I'm filtering it to just the genes from the cancer gene census. I'm often turning that off while exploring these data sets because then I can see an additional number of the genes throughout the human body. So across my 28 cases, that's crazy. Across my 28 cases, 19,000 genes are having various mutations in them. Now, the vast majority of those are modifier mutations, which are largely nothing. They may express things like transcription. They may be very impactful, but they're very hard to interpret. However, only a small number of those, relatively speaking, have a high impact from their mutation. And so if I select that, I can start to see that this gets rearranged. So a high impact, that's going to be something like a frame shift mutation or various other things. In fact, now that I've added that high impact mutation filter, which is only present across 11 of my APC genotyped cases, only 11 have that high impact mutation. And so that's going to be one of my cohorts. So this is actually pretty much done for one of my groups. So I am going to select that. I'm going to call this APC high impact. So just to show you some of the checks that I'm internally doing when I do that, I'm saying that, all right, well, these are high impacts. That means they should be comparable to each other. They've broken the protein. That's as opposed to something like a low impact, which is synonymous, so probably didn't even change a protein. An RNA transcription factor absolutely could be impacted by a low impact mutation, but for the most part, a protein coding RNA is not going to be changed at all by a low impact mutation. Moderate is going to be something like a missense, where a single codon or maybe even a couple of codons change. Those are going to be usually coded as moderate. But high impact, those are going to be things like truncations, where there's an early stop codon and the protein just stops being produced halfway through, omitting a big section, or frame shift mutation, which change a lot of amino acids and usually results in the protein not working. For the purposes of this class, will only accept high impact mutations being grouped together. If you're wanting to do a moderate mutation, you have to choose mutations that are comparable. And for that, you usually need to come to this mutations tab. And you need to go through and be like, okay, well, there are 13 people that have this specific mutation on chromosome 7 at position 140,753,336, where there's supposed to be an adenine, but there is a thymine instead. This is part of the BRAF protein. This is clearly an inherited mutation. How else are you getting the same nucleotide being mutated so commonly? This is where I said sometimes our inherited mutations are kind of coming through despite this masking that's been done. I believe that's because this is a inherited mutation in a cancer gene as opposed to a non-cancer gene, which might be more closely masked. I'm not really sure how their masking process worked. I looked at it, I couldn't really interpret their description on a cursory glance, but this would be a great opportunity for a project, looking at these 13 and comparing it to hopefully some cases that had no mutations in that gene. Not just cases that didn't have that mutation, but cases that had no mutations. Okay, so coming back to where we were at, we did make a cohort based off of high impact mutations in APC. However, I'm just checking my filter, so I'm just going to reset some of my filters here. I need something to compare those cases with high impact mutations to. And so to do that, I'm going to get any mutation. Please note there's no longer a filter here. And I'm going to select those 13 cases with any mutation. Any APC mutation. Now, there's something to note, is if there had been any C and V amplifications or any C and V deletions, those are going to be duplication events or deletion events, I would probably want to add those as well. Now, those, it turns out, can be a little bit tricky to add, and so oftentimes what I'll do is I'll just select over here and make those additional cohorts. However, that's not what happened in this particular case. And so I'm just going to now go over to the Analysis Center and do the set operations to subtract from my coad filter. Well, I'll check all three and we can kind of break that down and show how it gets interpreted. So here is my coad filter. Here's the 15 cases that lack any mutation. Here's the 2 cases that have a probably moderate or a low impact mutation within APC. And here are the 11 cases which have the high impact mutation, which of course is one of our types of any mutation. What I'm trying to do this for, though, is not to get that 2, it's to get these 15 cases that have no mutation. And so I'm now pretty much done. At this point, I would go on and start to get my sample sheets, but I'll be making a separate video for that, showing how we can get our sample sheets and how we can then filter them thereon. But here's my no APC. This is going to be one of my groups. This is probably the more wild type group. And then here's my high impact mutation. Now, one thing to just keep in mind before I head out is to remind you that these are still all tumors. So even if like our high impact, where we've kind of controlled for a particular type of mutation in APC, these are all still very mutated cells. These are still cancer cells. And so just keep that when you're in your head, which is a little bit hard to do. Keep that in your head as you're thinking about these things. Any comparison here is in order to compare cancer to cancer in a lot of these projects. You can do cancer to non-cancerous cells, usually of the same tissue type, but what you're going to get is there's massive differences, it turns out. It also turns out that getting those non-cancerous cell data can be a bit tricky because sometimes the system makes it kind of look like that data is available, but it turns out to not actually be available. But you can give it a try and see how it goes. Anyways, thank you and bye.